Hello everyone, welcome to the week 1 interactive session for classics in neuroscience. I am Ramya Shekhar, a PMRFTA for this course. Let me first begin by discussing the previous year assignment questions, followed by discussion of the topics that were covered in this week's lecture videos. First question: which historical period is highlighted for its contribution to modern neuroscience? So it is the 1950s and the 1940s and the 1950s, which are regarded as the golden period or the golden decade due to their contribution to modern neuroscience. And why is this the case? So let me go back to the text and discuss the same with you. So, if you see here, the 1940s and 1950s, especially in the 1950s, several discoveries were made across biology, and particularly in with regard to neuroscience, and many of them were extremely seminal discoveries, and some of these we'll be actually discussing as we go on in this course. For example, one of the very important discoveries in biology, that is the regarding the structure of DNA, which was discovered by Watson and Crick in their paper in 1953, in which they described a model for the double helical structure of DNA. That was something that was very important for the field of biology in general, but also had an important role in with regard to neuroscience. And several other discoveries that were made, and several other, for example, the Hodgkin and Huxley model, which we'll be discussing in the upcoming videos. Then regarding Renshaw inhibition, for example, then the fact that dendrites are actually active entities, they have certain active properties, and the invention of the intracellular electrode, which helped in giving us a lot of information about what happens within the neuronal cells, as opposed to the extracellular recordings that were being carried out previously. Then the idea of an action potential and how it works, and so on, and the synaptic pathways that are involved in the function of the spinal cord. So many such important discoveries and important ideas came forth during the 1940s and the 1950s. Hence, these are regarded as being this period is regarded as being very revolutionary in terms of the inventions and discoveries which propelled the field of neuroscience. So hence, the answer to this question is the 1940s and the 1950s. Next question: who is credited with laying the foundation for the chromosome theory of inheritance? So, although the chromosome theory of inheritance was proposed by Boveri and Sutton previously, they did not have any experimental evidence or proof for that. And so people were not willing to believe that basically. So, one of them actually was Thomas Hunt Morgan himself. He also was someone who actually did not believe strongly in this. But what happened was, in Hunt Morgan's lab, this is something we would have come across, when we must have read about genetics during our high school and so on, that in Thomas Hunt Morgan's lab, while his students were working with the fruit fly Drosophila melanogaster, they came across a white-eyed fly. So when they saw this white-eyed fly, so fruit flies generally have red eyes, but they came across this white eye fly, a mutant basically. So, Morgan wanted to understand that how is this, what is the nature of this inheritance of this white eye. And so when the white-eyed male fly was crossed to a was mated to a normal-eyed female, so what is expected and which also happened was that I mean not what is expected, as in what they observed was that all of the progeny, both male and female progeny, all of the progeny were red-eyed, which indicated that this white eye trait is recessive in nature, because the red eye was the dominant phenotype that was observed in the first filial generation. So all the progeny, both male and female, were red-eyed, which indicated that the trait responsible for the white eye is a recessive trait. Next, what happened was when they crossed a white-eyed female and a red-eyed male. So a normal male and a white-eyed female. What they saw was that while all the females had red eyes, all the males had white eyes. So they were not able to understand why there was this bias. Why is that only the males were having white eye, whereas the females were having red eyes? So basically, there was a sex linked inheritance. So wherever the it seemed as if this trait for the white eye was somehow linked to the X chromosome, which is responsible for the determination of the sex of the fly. So they said, so they understood, they realized, Morgan and his students, they realized that there is some kind of sex-linked inheritance, or there is something to do with the X chromosome. The inheritance of this trait is linked to the X chromosome. So, on the basis of this and several other experiments, similar kind of experiments, Morgan and his students, they came up with the idea that, they provided the strong confirmation that the chromosome theory of inheritance is actually true. That is, that genes are located on chromosomes. That is what the chromosome theory of inheritance states. And Morgan was the one who first provided the strong experimental confirmation of this chromosomal theory of inheritance by using his studies on the fruit fly Drosophila melanogaster. So, who is the one who is credited with laying the foundation for the chromosome theory of inheritance? Thomas Hunt Morgan. Next, which system is part of the sensory division of the nervous system? So, let me go back to the text. Yeah. So, the nervous system is divided into three great systems. And what are these systems? These are the sensory system, the motor system, and the central system. And a part of the sensory system is the is our ability to see our environment, or the ability of vision, and the others being smell, taste, touch, and hearing. So, which system is part of the sensory division of the nervous system? As we just discussed, vision is the correct answer. Vision is the one that helps us to see our environment, basically visualize the environment, and the information is then gets transmitted to the brain. And these others, so as you can see, the neuroendocrine system is part of the central system. Then higher motor centers, again part of the motor system, right? So, uh, the correct answer for this question is vision. I hope that is clear. Next, what structural feature of DNA did Watson and Crick describe? So, James Watson and Francis Crick were two scientists who were trying to come up with a model for the structure of DNA during the 1950s. So, there was this race to be the first one to uh, come up with a um, correct model for the structure of DNA. And several people were coming up with their own different types of models. So, Watson and Crick, they themselves had not been doing any experiments per se, but they were gathering information from the experiments that were being done by other people. For example, Watson and Crick interacted with Morris Wilkins, and Morris Wilkins shared with them crystallographic data that was collected by his colleague, who was Rosalind Franklin. So Rosalind Franklin and her student, they were collecting some crystallographic data for the DNA, the structure of the DNA. And Wilkins had shown Watson and Crick the crystallographic data that Rosalind had collected, but that was without the knowledge of Rosalind Franklin. And when they saw the that data, and also based on their several other in discussions and so on that they were had been having and with each other and with others, so they having put all of this together, Watson and Crick came up with a model for the structure of DNA. And what was the model? The model was that the DNA is a double helical structure. So double helical structure means it is a double helix. So there are this structure is what we call a helix, and it's a double helix because there are two strands, two complementary anti-parallel strands of the DNA molecule, which are wound around each other, if you can say in a way. So there are these two helical strands, so it's a double helix. They are in which there is complementary base pairing. And what I mean by complementary base pairing is that if on one strand there is an adenine, then on the other strand, which is anti-parallel to the first strand, on the other strand there is going to be a thymine. So A and T always pair with each other, and G and C always pair with each other. So there is always a complementary base pairing. So whenever there is an A on a strand, on the opposite strand there is going to be a complementary T. So if there is an A over here, this one is going to be a T. And if there is a T over here, the opposite strand is going to have an A. Similarly, if there is a G on a strand, on the opposite strand at the same position, there is going to be a C. So then these nitrogenous bases are going to have complementary base pairing. And this is going to be the sugar phosphate backbone. So as you can see, this is the sugar phosphate backbone, and the nitrogenous bases are projecting out of the backbone to form base pairing with each other by hydrogen bonding. So what structural feature of DNA did Watson and Crick describe? They described that DNA is a double helix which has complementary base pairing. Next, what is the primary role of RNA in protein synthesis? So, here you can see this large purple burger-like structure is the ribosome. And the ribosome is basically on the mRNA. So the ribosome passes along the mRNA and it tries to uh, read the mRNA. So the ribosome is responsible for helps in translating the information that is present in the mRNA. And how is that happening? So whenever where at the region where the ribosome is sitting on the mRNA, these tRNAs or the transfer RNAs are going to come and interact. So the mRNA is a messenger RNA, tRNA is transfer RNA. So at the region where the ribosome is interacting with the mRNA, the mRNA has codons. So codons are triplets of nucleotides. So these codons on the mRNA are they interact with corresponding anticodons on the tRNA. So the tRNA has this stem loop-like structure, and here are the anticodons. So depending upon the codon on the mRNA, the corresponding anticodon will interact with it. So that particular tRNA whose anticodon is able to successfully interact with this mRNA, that particular tRNA is going to have an amino acid attached to it. So you can see here that amino acid is attached to one end of the tRNA, and on this side you can see this is the anticodon. So when there is an interaction between the codon and the anticodon, there is also the amino acid that is present on the tRNA that goes and attaches to the growing protein. So here when the new tRNA comes and the amino acid on the incoming tRNA interacts, so there is a catalytic reaction. The ribosome has a catalytic region which can catalyze a peptide bond formation between the growing polypeptide chain and the amino acid of the incoming tRNA. So once this interaction happens, so the catalysis happens and this amino acid gets successfully incorporated into the growing polypeptide sequence, the tRNA leaves. So this is how information in the mRNA is translated into the formation of a nascent polypeptide. And that happens because this mRNA contains that information that is required for the formation of this protein. So depending upon the information that is present in the mRNA in form of the codons or the triplets of the bases, the the sequence of the polypeptide will be determined. So the mRNA acts as a template for protein synthesis. So the primary role of RNA in protein synthesis is to act as a template for protein synthesis. The other options here are incorrect because replication of DNA is a role that is played by the enzyme DNA polymerase. So DNA polymerase is responsible for is an enzyme that catalyzes the replication of DNA. Transporting oxygen is a function for proteins such as hemoglobin, and storing genetic material is the function of DNA itself. So DNA is deoxyribonucleic acid which stores genetic material and acts as a blueprint for the storage of information. Next question. What is the significance of the specific pairing in DNA proposed by Watson and Crick? Okay. So during DNA replication, what happens is that the double-stranded DNA, the helix that we discussed previously, the DNA double helix, there is hydrogen bonding between these nitrogenous bases, right? So these hydrogen bonds are broken. So during DNA replication, so these hydrogen bonds have to be broken and the helix has to open up. And when that happens, each of these strands, each of these original strands act as a template for the synthesis of a new complementary strand. And how is that possible? Because we know that there is complementary base pairing between the nitrogenous bases. So whenever there is a G on one strand, the opposite strand should automatically have a C. And whenever there is an A on a strand, the opposite strand should automatically have a T. Therefore, when these when the original DNA double helix is the hydrogen bonds are broken and the helix opens up, the complementary strand can automatically be synthesized based on the information that is present in the original parental strand. So, because there is a G on the original parental strand, we know that the new complementary strand should have a C on over here. And similarly, when the parental strand has an A on that at a particular position, automatically the complementary strand should have a T on at that position, and so on. And therefore both the parental strands can act as templates for the synthesis of complementary strands. And finally, what happens is that at the end of replication, you see that there are two identical double helices. Each has one old strand and one new strand. This one also has one old strand and one new strand. So because of the presence of complementary base pairing in DNA, this is possible. Okay, so based on this explanation, what is the significance of the specific pairing in DNA proposed by Watson and Crick? So the correct answer is it suggests a possible copying mechanism for genetic material. So that is what we just saw. That because of the presence of the specific complementary base pairing, it suggests that, and it actually is the way it works, is that there is the ability to synthesize a complementary strand and therefore allow the copying of the DNA. So each of these strands can then go into different daughter cells. Next question. Which statement aligns with the central dogma hypothesis? So what is the central dogma hypothesis? What does the central dogma hypothesis suggest? So the central dogma hypothesis suggests that the transfer of information takes place from DNA to RNA via transcription. The information in the DNA is copied like we just discussed, or it is replicated via the process of replication. So that allows the inheritance of the information that is present in the DNA. So replication is the process by which the DNA gets copied. So to produce two identical strands of two identical DNA which have the same information as the original DNA molecule. That is the process of replication. And then the information in the DNA is transferred from DNA to RNA by the process of transcription. So in DNA is the material of the hereditary material, or the is the material is the molecule or the entity that stores the genetic information. It acts as a blueprint for genetic information. Whereas this information that is there in the DNA, that is transcribed into RNA. So RNA acts as a messenger of the information that is present in the DNA. So RNA takes that information that is there in the DNA for the formation of functional products. So DNA to RNA happens by transcription. Then from RNA to protein, the information is like the process of transfer of information from RNA to protein happens via translation. So we just discussed a little while back about how translation happens and about how RNA acts as a template for translation. And then in some cases, uh, especially, I mean, in, for example, in case of viral, in viruses and so on, what happens is there might be reverse transcription also. So reverse transcription is the transfer of information from RNA to DNA. This is not a very common, I mean, as in, it is not something that happens in all cells. So reverse transcription is a very restricted, it's relatively restricted. So reverse transcription is basically from RNA back to DNA. So the central dogma says that information flows from DNA to RNA to proteins, and then it cannot reverse back from proteins to RNA to DNA. So that does not happen. The reverse transfer of information from protein to RNA to DNA does not happen. So the information happens, the transfer of information happens from DNA to RNA to protein. And why is that? Because proteins generally, after their functions are in the cell are completed, they undergo degradation. So proteins are under constant turnover. So there isn't any transfer of information from protein to RNA to DNA. So, which statement aligns with the central dogma hypothesis? First, information flows from RNA to DNA and then to proteins. Of course not. Information does not flow from RNA to DNA to proteins. It flows from DNA to RNA to proteins. The second option, information flows from DNA to RNA to proteins and cannot reverse. Yes, that is the correct answer. And the third and fourth options, information flows only from proteins to DNA. Of course not. Information does not flow from proteins to DNA. DNA does not participate in information transfer. That is absolutely wrong because DNA is the one that is required in the first place for the transfer of information. Because DNA is the one that actually contains the information that is required for the synthesis of the RNA and the protein. Next, what discovery was made by Hershey and Chase in 1952? Okay, so what was the experiment that Hershey and Chase had actually performed? Okay, so this is the famous Hershey and Chase experiment for which Hershey actually got the Nobel Prize. I mean, he shared the Nobel Prize with Max Delbruck. But yeah, this was the famous Hershey and Chase experiment, which showed that it is indeed DNA, which is the genetic material. Because up until that point, many people, there was not many, I mean, there was still a few people who were not willing to accept that DNA is the genetic material. They were, in fact, many people were saying that DNA might just be the function of DNA might be that it's just for the storage of phosphorus. And many people, and they were saying that DNA is not complex enough for storing information, and it is probably proteins which are actually the entities responsible for storing genetic information. This was something that was actually this idea was actually something that many people believed in, and people were not very easily willing to accept that it is DNA which is the genetic material. So at that time, what Hershey and Chase did was that Martha Chase and Hershey, these two people, they performed this experiment using bacteriophages. So bacteriophages are basically viruses that can infect bacteria. So these are basically a type of virus whose host are bacteria. And the bacteriophages that they used are the ones that will transfer their genetic information. So basically, the way it works is that bacteriophages first infect the bacteria. So basically, they go and attach to the cell wall. So this is called adsorption. So they get adsorbed to the cell wall, and this is when they inject their genetic material into the bacterial cell. And then when they after they have injected the material into the bacterial cell, the genetic material, then there is propagation of the genetic material propagates within the bacterial cell, and then the bacteriophage basically they may lyse the cell, and they multiple of mul I mean because after they have genetic material has multiplied, oftentimes they may undergo a lytic process, and the bacterial cell is lysed and the bacteriophages are released. So this is how bacteriophages often work. They may work by lytic or lysogenic processes and so on. So, but the initial steps involve the basically the adsorption of the bacteriophage, that is in which they go and attach to the cell wall of the bacteria, and then they inject the genetic material. So basically, they're infecting the bacteria. So, using this information, and because they wanted to determine whether it was proteins or whether it was DNA, which was the genetic material, what they did was they used sulfur labeled, they used radioactive sulfur. So the radioactive sulfur will label the capsule of the bacteriophage. So bacteriophage have only two components in them. One is the capsule of the bacteriophage, which is made of proteins, and the other is the interior. In the interior of this capsule is the DNA. And the DNA is, we discussed what is the structure of the DNA and so on, right? So they wanted to know whether it's the protein or the DNA, which is the genetic material. So with whatever is injected into the cell base and causes the infection by getting multiplied is the component that is the genetic material. As simple as that. So they used radioactive sulfur, which will label the capsule of the bacteriophage, the protein which is made of protein. And they used radioactive phosphorus, which will label the DNA. And then they allowed such bacteriophages to infect the bacterial cells. So when these bacteriophages infect the cell, they use very high speed blending. So basically, this process basically will cause the the capsule to come off. It will basically remove the bacteriophage which has been adsorbed onto the cell. It will basically remove them. And then after, so basically whatever this attached capsule gets removed. And then after centrifugation, they will detect whether this bacterial cell has radioactive sulfur or radioactive phosphorus. If there is radioactive sulfur, that indicates that it was the protein which got injected into the bacterial cell, which has entered the bacterial cell, and that is the genetic material. If there is radioactive phosphorus, that indicates that it is the DNA which has entered the bacterial cell, and the DNA is the genetic material, because that, I mean, whatever, like we discussed, whatever enters the cell and undergoes, whatever is replicated and results in the formation of the new bacteriophages, that is the genetic material. So they saw that they were not able to detect the radioactive sulfur in the cells after the blending or the centrifugation, but they were able to detect radioactive phosphorus. And this indicated that it is indeed DNA which is getting injected into the cell. And that was the one responsible for the infection. And therefore, it is DNA which is the genetic material and not proteins. So, this was the experiment which uh, established in the field that DNA is the genetic material. Coming back to the question, what discovery was made by Hershey and Chase in 1952? The correct answer is that DNA is the genetic material. Next question. What is one structural limitation of ribose sugar that prevents its use in DNA? So, what is the difference between the ribose sugar and the deoxyribose sugar? First of all, we know that it is the deoxyribose that is present in the DNA. So, the full form of DNA is deoxyribonucleic acid, right? And the deoxyribose sugar is the that is the component of DNA, and the other sugar, that is ribose, is not a component of DNA, rather it is a component of RNA. So RNA is ribonucleic acid and ribose is the component of RNA. What is the difference between deoxyribose and ribose? Ribose has an OH in the attached to the 2' carbon of the ribose sugar, whereas in deoxyribose the 2' carbon has only an H attached to it. So there is an extra oxygen that is present in at the 2' attached to the 2' carbon in case of the ribose sugar. First of all, the presence of this oxygen causes the relative instability of so RNA, like we discussed, RNA, the component ribose is a component of RNA, and the presence of this oxygen in fact makes the RNA more unstable relative to DNA, because this OH is prone to undergo hydrolysis. So, just a minute. Yeah. So, yeah, like we were discussing, this, first of all, this OH makes RNA relatively more unstable as compared to DNA, because it makes it more prone to undergo hydrolysis. The second thing is that this extra oxygen atom that is present in the ribose sugar would also create unfavorable van der Waals interactions. And these unfavorable van der Waals interactions would basically disrupt the formation of the DNA double helix. So that's why the presence of this oxygen would hinder the formation of a proper double helix, and that's why there is only deoxyribose that is present in the DNA. I hope that is clear to you. So, what is the structural limitation of ribose sugar that prevents its use in DNA? The structural limitation is the presence of this 2' oxygen. Presence of this oxygen at the 2' carbon of the ribose sugar that one creates these unfavorable van der Waals contacts with other creates these unfavorable van der Waals contacts, and second is that this oxygen also makes it more unstable. Sorry. Next, what genetic disorder is caused by a defect in the gene for hemoglobin? So, hemoglobin is a protein that is present in red blood cells and it is responsible for the transport of oxygen. So we know that our body requires oxygen, and whenever we breathe in the inspired air, whatever oxygen is there, that oxygen is transferred at the alveoli to the blood, and in the blood this oxygen binds to the or is transport is interacts with and is binds to the protein called hemoglobin that is present in our red blood cells. So this hemoglobin is required for the transport of oxygen to the different tissues of the body, which require the oxygen for respiration. But what happens is, in case there is a mutation in this protein called hemoglobin, there, especially a particular point mutation in hemoglobin, what happens is that this abnormal hemoglobin forms strands within the red blood cells, and these strands cause the shape of the red blood cell to change. So normally red blood cells have a biconcave shape when the hemoglobin is also normal. However, when this there is a mutation in hemoglobin and which causes the hemoglobin to become abnormal, this hemoglobin forms strands which result in the change in shape of the red blood cells themselves, and these red blood cells become sickle shaped. So rather than being the biconcave shape that they are usually supposed to be, they become like a sickle. So that is why this disease that comes about because of these sickle shaped red blood cells is called sickle cell anemia or sickle cell disease, because these sickle cells, so the normal red blood cells are able to easily flow and move along the capillaries of the capillaries and carry the oxygen to the different tissues. But when these RBCs become sickle shaped, what happens is that they end up, so they become sticky and they get clogged in the capillaries, and because of them getting clogged in the capillaries, they block the blood flow itself. So not only is there a problem in the transport of the oxygen, because of the abnormality of the hemoglobin structure, because of the formation of these sickle shaped cells, the blood flow itself also ends up getting blocked. So, because of that, there is a severe lack of oxygen to the different tissues, and this is called anemia. So, this disease is called sickle cell anemia because of the sickle shape of the that the RBCs attain, and the anemia which results because of the lack of oxygen to the different tissues of the body because of a mutation or a defect in the gene for hemoglobin. Next, the neuroscience program was formed by. So this was something that was discussed in the video lectures, right? It was Francis Schmidt who actually came up with a formal neuroscience program, and this happened in the year 1960. Next question. According to the hierarchical levels of organization, which is the correct order? Okay, so let me just go back to the text. So these are the different hierarchical levels of organization. So it starts from the lowest level, you can say, is the genes. So genes are basically they are fragments of DNA, or they are parts of DNA which can code for a functional product. So this functional product might be an RNA, or it might be the end functional product might be a protein. So the first level is the level of the genes. Then comes the level of molecules. So molecules can be proteins, can be carbohydrates, can be several different kinds of molecules that carry out various functions in the cell. So different molecules have very different functions depending upon their structure and their sequence and so on. So the genes are we discussed that genes are present on chromosomes. Molecules are entities that are present throughout the cell, and they carry out different functions. Next comes synapses. So synapses is basically are the connections or the contacts between different cells with each other. Specifically, in terms of the neuronal cells, we know that neurons contact each other via the formation of synapses. These might be chemical synapses, or these might be electrical synapses. Then comes the level of circuits. So synapses are when individual neurons are connecting with each other. Circuits is the it's a an even higher level phenomenon in which there are multiple neurons that are all interconnected with each other, and it basically they are all parts of different specific systems. And then beyond that comes the higher cognitive and social functions that are basically, you can say, emergent properties of these circuits. So these are the different hierarchical level of organization. So now coming back to the question, according to the hierarchical levels of organization, what is the correct order? So the correct order starts with, let us go through the options. First option is higher cognitive function, higher order cognitive function, then molecules, then synapses. No, we know that this is not the correct order, because higher order cognitive function is the highest level of organization, and molecules and synapses are so higher order cognitive function is the highest, synapses is somewhere along the middle, and molecules is lower than that. Next, genes, higher order social functions, molecules. No, we know that genes are the lowest level, then comes molecule, and higher order social functions are at a much higher level of organization. Third, synapses, genes, circuits of specific systems. Again, genes are at the lowest level, then comes molecules, then comes synapses, then comes circuits, and then finally comes the higher order cognitive functions. So the correct option is genes, followed by molecules, followed by synapses between two different cells, basically. Next, who proposed the term genetics as a specific biological term for the study of the roles of heredity? So, the person who proposed this term was William Bateson. And just in case you don't get confused, Gregor Mendel did not come up with the term genetics. In fact, during his study, he did not use the term genes. He had used the term independent factors. So during Mendel's time, uh, the term genetics was not used. It was William Bateson who coined the term genetics. Next, who demonstrated that mutations could be induced by X-rays in 1927? So this was done by Hermann Muller. So Hermann Muller received the Nobel Prize for this particular discovery that X-ray mutations can actually, so X-rays can actually induce mutations. So as you can, this is Hermann Muller, who is actually work. You can see all of these bottles and vials of uh, fruit flies that he is working with, and here also he's looking at a vial of fruit flies using this particular instrument. So Hermann Muller actually used fruit flies or the Drosophila melanogaster as a model, and he showed that by using different dosage of X-rays that these X-rays can actually induce mutations. So Hermann Muller was actually he was basically he had become he had a good bonding with the students that who were there in Thomas Hunt Morgan's lab, and slowly he also formally became a part of the lab's discussions and experiments and so on, and then later he moved elsewhere, and he pursued his own independent line of work and all of that, but he inspired by his in I mean as in because of his training and interaction with uh, Thomas Hunt Morgan's lab who were working on Drosophila. So he also used Drosophila to study the effect of X-rays, and he discovered that X-ray radiation actually causes mutation or a change in the DNA. So the term mutation means a change in the change at the level of the DNA. So next question, which of the following statements about cellular biology is correct? Okay, so in eukaryotes, we know that eukaryotes have a nucleus, whereas prokaryotes do not have a nucleus, right? Prokaryotes generally only have a nucleoid. They don't have a membrane-bound nucleus per se, right? So in eukaryotes who have a cell nucleus, which is a double membrane-bound organelle, the DNA is present within the nucleus. As you can see, this is the cell nucleus. The DNA is present within the nucleus, and the RNA is getting transcribed within the nucleus. So the process of transcription in eukaryotes happens within the nucleus. And then this RNA undergoes various kinds of processing. And then this processed mRNA is transported to the cytosol basically. So it is exported via these nuclear pore complexes. So, and I mean, as in there are nuclear pores over here, and through these nuclear pores, this processed mRNA is transported. And once this mRNA enters the cytosol from the nucleus, this RNA is this I mean the ribosomes basically get loaded onto the mRNA. So these ribosomes when they interact with or they get loaded on the mRNA, what they do is, like we discussed, these ribosomes travel along the length of the mRNA, and the tRNAs get the based on the codon-anticodon interaction between the mRNA and the tRNA, the amino acids are incorporated into the growing polyprotein chain. And then this protein chain finally leaves the this particular complex. So this process is called translation. So the process of the generation of the protein from the mRNA is called translation, and this happens in the cytosol, where the ribosomal machinery is present and the tRNAs are also present. Whereas transcription happens in the nucleus, where the DNA is present. So this is how the transcription and translation are happen independently in eukaryotes, whereas in prokaryotes, transcription and translation are coupled events. So, which of the following statements about cellular biology is correct? First option, DNA replication occurs in the cytoplasm, whereas protein synthesis occurs in the nucleus. No, because DNA replication happens in the nucleus, whereas protein synthesis happens in the cytoplasm. Next, both DNA replication and protein synthesis occur in the cell nucleus. Of course not. Only DNA replication happens in the nucleus, whereas protein synthesis happens in the cytoplasm. Third, DNA resides in the cytoplasm, whereas proteins are synthesized in the cell nucleus. It is the other way around. DNA resides in the cell nucleus, whereas proteins are synthesized in the cytoplasm, which is the fourth option. So that is the correct option. Next, according to the central dogma hypothesis, what happens once information has passed into protein? So this is something we had discussed, that the information that is passed into protein cannot be reversed back into DNA, and it also does not remain indefinitely in the protein, nor does it get transferred to RNA. And why is that? It is because it is lost irreversibly, because proteins undergo turnover, or proteins are degraded by the different systems in the cell, for example, the ubiquitin proteasome system or the autophagy system, and whatever, depending upon the state of the protein undergoes turnover after its biological function is complete. So the information cannot be reversed back into DNA once it has passed into protein. It also does not remain indefinitely in the protein. It also does not get transferred to RNA. The correct option is that it is lost irreversibly. Next, what substance initially observed in glandular cell cytoplasm by Garnier around 1900 was later recognized as crucial for protein synthesis by the early 1940s? So this substance, which Garnier first observed in the glandular cell cytoplasm, was basically RNA. And later, in the early 1940s, it was recognized that this RNA, which is a single-stranded structure, is actually crucial for protein synthesis. So as many of you would know, RNA is a single helix. It is not a double helix. DNA is a double helix. RNA is a single, like it is a single stranded entity. DNA has two strands. That's why it's a double helix. However, RNA can form secondary structures by interacting within itself. So RNA can interact with itself to form secondary structures. Otherwise, RNA is actually a single helix or a single stranded entity. And another difference between RNA and DNA is that RNA, the nitrogenous bases that are present, adenine, guanine, and cytosine are common with DNA, but uracil is present only in RNA and is not present in DNA. Similarly, thymine is present only in DNA. It is not present in RNA. Uracil actually has a structure that is quite similar to thymine. So wherever thymine was present in DNA during transcription, wherever there is an A present in the DNA during transcription, the complementary strand on the RNA would have an uracil rather than a thymine. And the other difference between RNA and DNA is that RNA is ribonucleic acid. It has the ribose sugar, whereas DNA is deoxyribonucleic acid. DNA has deoxyribose sugar. Next question. What chromosomal abnormality is associated with sickle cell anemia? So we discussed that it is the hemoglobin gene or the gene for that encodes the protein hemoglobin that might have a defect. So what exactly happens is that this hemoglobin gene at the sixth amino acid position, right? So at the sixth amino acid position where there is supposed to be a glutamic acid, okay, as in the sixth amino acid position is that which encodes for, which contains a glutamic acid residue. And this is encoded by the following, the corresponding uh, uh, region in the DNA, it should have a this particular sequence. But if there is a point mutation in which happens in certain entities, which results in the so there is a point mutation, as in a single nucleotide has been changed. So there should be a T and an A over here. And what has happened is there has been a point mutation such that it has become an A over here. So it should be a CTC. It has become a CAC. So on the opposite strand, naturally, it instead of being a GAG, it has become a GTG. And because of that, what happens is instead of encoding a glutamic acid, what happens is that the amino acid residue that gets incorporated is a valine. So instead of glutamic acid, which is an acidic residue, what it gets encoded is valine. So there is a point mutation which results in the formation of this mutant hemoglobin protein. And because of this mutant hemoglobin protein, this hemoglobin that is supposed to be formed. So hemoglobin has a quaternary structure in which there are four different polypeptide strands. So there's an alpha, alpha, and beta. So what happens is that there is a the beta strand basically is the mutant one, and what happens is because of this mutation the hemoglobin structure is no longer the way it should be. It, like we discussed, this hemoglobin is not able to interact with oxygen like the wild type hemoglobin is able to. And because the it's an inability to interact with oxygen, as well as the this hemoglobin also does not stay in its natural shape. And because of that, what happens is that the biconcave RBCs no longer stay that way, they become sickle shaped. So there is a point mutation in the gene for hemoglobin that in the at the level of the in the chromosome basically, and that causes the disease sickle cell anemia. Next, what method was used by Okowa and his colleagues in 1955 to generate synthetic RNA? So Severo Okowa, who was a scientist who received the Nobel Prize in Physiology or Medicine in the year 1959 for his discovery of this enzyme called polynucleotide phosphorylase. So, I mean, he named it as polynucleotide phosphorylase, and in vitro, this enzyme was able to allow them to synthesize or generate synthetic RNA. Although in vivo its function is the opposite, its function is to degrade the RNA. But in vitro or in the test tube, this enzyme that he coined as polynucleotide phosphorylase was able to allow them to generate synthetic RNA. And using synthetic RNA, this was the first time synthetic RNA, I mean RNA, they were able, the scientists were able to synthesize RNA by themselves. I mean, as in to generate synthetic RNA, this was the first time. So because of, because of this discovery of polynucleotide phosphorylase, Okoa and his colleagues, so Okoa received the Nobel Prize in Physiology or Medicine. Final question. Which enzyme catalyzes the synthesis of DNA in Escherichia coli? So we know that the process of synthesis of DNA or the copying of DNA is called as DNA replication. And in E. coli, like I discussed, the process of because E. coli do not have any nucleus as such, the DNA is present in the cytoplasm. So the replication also happens in the cytoplasm. So, in DNA replication, the enzyme that is responsible is the DNA polymerase. So, E. coli have DNA polymerase 1, 2, and 3, and it is generally 1 and 3 which play a major role during the replication. So there is an origin of replication, and the direction of replication is bidirectional, because at this origin, when the replication begins, the replication happens in both this direction and this direction. So the origin of replication is bidirectional, and there is a leading strand, and then there is a lagging strand, or also called the Okazaki fragment. So the leading strand undergoes continuous synthesis, whereas the lagging strand undergoes discontinuous synthesis. So these discontinuously synthesized fragments are called the Okazaki fragments. And the replication happens, like I said, because of the enzyme DNA polymerase. And as its name suggests, DNA polymerase synthesizes the new strand of the complementary strand of the DNA based on the because of the, like we discussed, because DNA has complementary base pairing, using one of the strands of DNA as a template, the DNA polymerase is able to synthesize the complementary strand. So let me discuss some of the concepts that were discussed in this week's video lectures, and then we can close the meeting. In case you have any doubts regarding any of the assignment questions that were discussed, or if you have anything that you want to ask or share, feel free to drop your questions in the discussion forum. I'll be happy to answer you. So, what were some of the key uh, topics or key things that were discussed in this week's video lectures? So, first is that the point that the study of the nervous system, that is neuroscience, uh, this came about. I mean, as in the in history, the idea that we should be studying neuroscience separately did not exist for a long time. The nervous system was actually studied as a part of several other systems, such as it was a part of neuroanatomy, neurophysiology, pharmacology, neurology, psychiatry. So these are all different fields of medicine, and there was no formal discipline of neuroscience as such for a long time. And it was only in 1960 that Francis Schmidt actually came up with the idea that there should be a neuroscience program. So, this idea that we should be studying the nervous system in greater detail is something that actually the idea that there should be a formal discipline, that was something that actually helped propel the field of neuroscience greatly. Why are we so interested in studying the history of neuroscience? Is because we need to understand that currently there is this many people who have this mindset that the whatever was done previously, or the old literature and the old experiments may not be so relevant or so important, is a belief that many upcoming scientists might wrongly have. And that is actually not correct, because it is the reason that we are able to currently have this current knowledge and understanding of neuroscience is because of the work done by all of those individuals at that particular time. So, because of the experiments that they did, and because of the information that they gleaned, and because of the observations they made, and by building upon that, the different people building upon the knowledge and the information that was gained by others, and that is how the field has progressed to its current extent. So always remember that you're always standing on the shoulders of giants whenever you are actually using the existing knowledge and building upon it. It is because that knowledge actually exists, because of the work of people before you, is why you are able to progress even further. So, the history of neuroscience will actually tell us the thought process of those individuals, and the way those individuals approached the questions in the field, and what were the level of creativity that they employed to actually come up with the observations that they did, and the hypothesis that they built, and the rigorous nature of the experiments that they performed, the meticulous nature by which they actually carried out such experiments using relatively uh, primitive or relatively less fancy equipments as compared to the ones that we currently use. So, although you might think that it's not as relevant in terms of the equipment and so on, it is actually relevant in terms of the way they actually addressed the problems at hand. and how they came up with the observations they did, and what we can learn from their approach, their determination, and the way they navigated science in general. So, the history of neuroscience, like we discussed, embraces many disciplines. It was not a formal discipline by itself, it was actually studied as a part of several disciplines. And even now, we understand that neuroscience is a very interdisciplinary field. It encompasses and interacts with several other fields, and and this interdisciplinary nature of neuroscience has actually, is also is it's both its charm and also introduces the complexities that are associated with this field. So, next, the history of neuroscience extends across many species, it extends across all systems, and extends across all hierarchical levels of organization. So, what do we mean by that? It extends across all species. So, the biological world that we are studying, it is, let us remember that we are studying it in the context of the evolutionary theory. So we know that uh, over the course of evolution, the organisms have evolved from the relatively less complex bacteria and single-celled and lower multicellular organisms and so on, to the animal kingdom, in which starting with the invertebrates, in which compose of the arthropods and insects and mollusks and so on, and then in the vertebrates you have the fish, amphibians, reptiles, birds, and mammals. And then in the mammals you have the several other mammals, and then you have the non-human primates such as the chimpanzees and so on, and then you have the humans. So this is how, we can see that across evolution, even from even invertebrates such as insects and arthropods and mollusks have a nervous system. Although relatively primitive compared to our nervous system, they do have a well functioning nervous system. And several insights in neuroscience, even many classic, many path breaking insights that we currently have regarding the field, have come from studies that have happened in these other organisms. For example, we know that the squid giant axons were used by Hodgkin and Huxley for their famous experiments. Then several people have used Limulus polyphemus, which is the horseshoe crab, for several experiments. And Aplysia is also a favorite model, which has given us several insights. And insects like Drosophila melanogaster have continued to be used as models across the world to study diseases such as neurodegeneration and cancer and neuroinflammation and so on. Even worms, nematode worms like the C. elegans, have told us, have given us insights about the olfactory system and circuits, neuronal circuits, and so on. And invertebrates, of course, several studies in cats and cats have, we'll come across those in future video lectures about experiments that were done with cats, the nervous system of cats, and even now experiments are being done in rats and mice, in those are like those are standard model organisms that are used not just in the neuroscience but in also in other aspects of biology. And similarly, animals like chimpanzees continue to be used for both behavioral experiments as well as experiments concerning the neurobiology of how thing I mean the new both in terms of molecular and cellular neuroscience as well as in terms of behavioral neuroscience. So, and of course, amphibians like frogs and newts continue to be, have been used historically, and even now, certain labs do use them for increasing our understanding in various aspects of the field. Then, the history of neuroscience extends across all systems. So, we discussed that the nervous system is divided into three different great systems. The sensory system, which are responsible for receiving the information from our surroundings, basically, in us, receiving the different kinds of information, such as the smell, taste, our sense of touch, our sense of hearing, our ability to see our environment, and so on. So, these are all part of the sensory system in which we obtain the information from the environment. Then comes the motor systems in which we react to the information that we have received from the environment. So whatever the sensory information we have received, and it undergoes processing in the processing in our nervous system, and finally nervous system decides that we should react in a certain way. So all of that comes in the purview of the motor systems. So this consists of the autonomic nervous system, the nervous system, the systems responsible for posture, for reflex actions, for central pattern generation, for the spinal cord synaptic pathways, and the higher motor centers. Then come the central systems. So central systems comprise the neuroendocrine pathway, then circadian rhythms, then feeding, mating, reproduction, and so on. So these are the three major systems that neuroscience, the nervous system is divided into, and the history of neuroscience actually extends across all of these systems. So we will be discussing about work that was done with regard to vision, with regard to our sense of touch responses, and with regard to our sense of olfaction, with regard to the reflex actions, with regard to how the synaptic pathways of the spinal cord work, how the motor centers, are interacting with the muscles and the glands and so on. So we will be discuss and how the central systems, what kind of how is learning and memory established, and so on. So we will be touching upon all of these in different ways in the upcoming video lectures. Next, the history of neuroscience extends across all hierarchical levels of organization. So we have already discussed that what are the different hierarchical levels of organization. It extends from the lowest level, which is the genes, to the molecules, to the synapses, to the circuits of different specific systems, and finally to the higher cognitive and social functions. This is the highest level of hierarchy, and each of these levels, you can imagine the like the the level above it is actually because of the emergent properties arising from the interaction between the lower levels. So because of the interaction of several synapses with each other, the circuits are established. Because of the interaction of different molecules with each other, the synapses are established, and so on. So the each level of hierarchy, each level of organization is actually arising because of the emergent property, arising because of the as an emergent property of the interactions that are happening at the previous level of organization. And all of these levels are important because defects that occur in each of these levels can result in different types of disorders. For example, defects at the level of genes or mutations in genes can cause genetic disorders. So we know that there are several neurological disorders which have a strong genetic basis. So mutations in genes can cause different kind of neurogenetic disorders, neurological disorders, or neurodevelopmental disorders. Then if there is an aberrant interaction between the molecules, or if there is a loss of interaction between molecules, that can also result in different kinds of imbalances and disorders and so on. At the level of synapses, if there is any if there is the synapses are not firing properly, or if there is an imbalance in terms of the excitation and inhibition and so on, or if the synaptic architecture is hampered, or if the synaptic function is hampered, because of that also several diseases can happen. Then in terms of circuits, at the circuit level, if there are the circuits are not correctly wired, or if there is any if there is a problem in at depend on the which circuit is active at what time and so on, that can also lead to different kind of disorders which fall under the purview of the departments of psychiatry and neurosurgery and neurology in the clinical spectrum. Then, if the higher cognitive and social functions are hampered, then of course you see that at the behavioral level, you see that there are certain problems that arise. So we need to understand that neuroscience by itself also has these different hierarchical levels of organization. And the history of neuroscience, you'll see that it has been studied at all these different levels of organization. So there have been studies happening and that have happened at the level of the genes and molecules. There are also studies that have happened at the level of synapses, and those that have happened at the levels of circuits and behaviors and so on. So there are the at the level of history also, you will see that different people have tried to address the different levels of different hierarchy, different levels of this hierarchy by performing different kinds of very ingenious experiments. So next, what are the factors that produce discoveries? So there are 6 critical factors. These are methodology, the biological preparation, the principal investigator, the theoretical framework under in which you're with which you're working, of course, a lot of chance, and different kinds of support. So support can be financial or institutional, or support by your family, support in terms of the support that is required with your by your collaborators, by your students, by your PI, and so on. So these factors are actually very important for any great discovery in science. So the methodology might need not be very fancy and so on. It may not be, you need not require the most newest, coolest experimental setup. But if you have, like as you will see in upcoming videos, as you will see that if you have a very elegant setup, elegant experiment which relies on the most practical and the most frugal setup that you can use, the but it gives you the required results. That is the best equipment that you can have. That is the best methodology you can employ. The biological preparation of course, what kind of model system are you using? Are you using, I mean, like, different models have their own advantages and disadvantages. There are certain models which are relatively cheaper to handle, and there are certain models which are more complex to handle, but they'll also give you a lot of information. There are those in which they may not be very easy to handle, but they might have some other advantage. They might have some setup which makes it easier for you to address the question that you want to answer. They might have certain behaviors that you wish to understand. So different model systems have their pros and cons, and it is important to choose your model system based on the kind of question you are trying to ask and its suitability and your accessibility to that model system. Next comes the investigator. Different investigators have their own approach, have their own background, have their own understanding regarding a subject, have their own ideas and hypotheses. Their mindset also matters, whether they are interested in collaborating with other individuals, whether they are open-minded, whether they have a fixed mindset regarding their observations, whether they are willing to listen to other people's point of view, whether they have the drive and the initiative to keep pursuing an experiment, no matter how many times it fails, whether they are able to come up with the required funding to run the laboratory, whether they are jovial or whether they are very moody, whether they have a good relationship with their students and their peers and their mentors, and so on. Lots of factors are involved. Next comes the theoretical framework. How well do you understand the theoretical framework with which you're working, and how much you're restricted by that, and are you taking that into consideration properly while you're doing the experiment? Next comes, of course, chance. That is not something uh, that is quite literally out of our control. Then comes support. So, of course, if any laboratory requires good financial and institutional support, and all the individuals working in a laboratory, of course, require cooperation within each other, between each other, and the support of their families and peers. So, these are some of the important factors that uh, may play a very critical role in propelling discoveries in general. Not this is something that is not is true not just for neuroscience. It is something that is true for science in general. Of course, just like that is true for science in general, these ethical issues that are being discussed and are also true for science in general. So, the ethics of any research is extremely important. So, there are, in history, there have been several relatively, current, if you look at it with the current point of view, though there have been experiments that have been conducted that might be in the gray zone, or they might be completely unethical with regard to current ethical standards, because ethical standards are ever evolving, and of course we can't judge the experiments and the methodology that were used at that time using current uh, lenses, but we should ensure that we do not propagate that. We should be more conscious that the currently what we are doing, it should at least meet the ethical standards that are currently in place, because ethical standards continue to evolve with time. And history has shown us that there have been lots of ethical issues that have cropped up because of all of these different factors. There have been scientists who have been ignored or whose work has not was not very well known because of their because of the fact that they published their papers in their own native languages such as say French or German, and it was not because uh, English is often regarded as the language of science, and most papers are published in English, and people across the world continue to publish in English. So, the papers that were published in other languages did not receive the same level of appreciation or and the outreach that papers that were published in English had received. And several scientists have, of course, their contributions have therefore remained forgotten or were not recognized to the extent that they actually deserve to be recognized because of that. Then comes the political intrusion and coercion. So, as you will see in future videos, a lot of these experiments that we'll be discussing happened during wartime, especially World War II. And we know that during World War II, there was a persecution of Jewish scientists in Nazi Germany, and many of them had to flee from. Uh, their native land because of the World War and had to seek refuge in other countries and carry out their experiments in a great deal of excuse me, yeah, they had to carry out their experiments in very difficult times. So of course, then about the role of bias on the basis of race and ethnic groups. So many people have faced a lot of persecution because of their race. For example, Jewish scientists have during because like you discussed during World War II, many Jewish scientists were not allowed to publish their work in German journals because of their Jewish ethnicity. Then black people and brown people, even I mean, black people have faced a lot of persecution and a lot of struggles because of their black, I mean, being a black person, and this is true even for people from uh, people who are brown. Then recognition between single discipline and multidisciplinary studies. Of course, there are neuroscience, like we discussed, is a lot of neuroscience involves very many involves a multidisciplinary approach, and that means that these kind of studies might also be more complicated to execute, would require collaboration between several different kinds of individuals, and that will of course result in more complex and more complicated endeavors. So, it is important to understand that neuroscience is such a discipline, and it requires, in many cases, a more collaborative ventures between different laboratories. Then comes recognition and competition between laboratories. So, like we discussed regarding the model of the double helix, there was a competition, right? Or there was a race. So this idea of competition between laboratories and the idea that your work might be scooped, or the idea that publish or perish, these kind of things do exist. This is true for science in general, the academia in general, although it is, I mean, um, competition, as long as it is healthy, as long as it encourages discussions, as long as it encourages knowledge, and it encourages the progress of the field, is good. It should not become so cutthroat that the field loses out on the progress that should happen. Next comes recognition between male and female. So in several cases, male and female, there's the gender bias between male and female colleagues has caused a lot of it has it has been very apparent in several cases. For example, in case of Hershey and Chase, so Martha Chase faced a lot of discrimination. She found it very difficult after that her work with Hershey Chase when she went on to marry, and then when she went on to try to find positions, for example, she found it very difficult. It was not easy for her to find positions because she was a female. Several other female scientists have expressed the fact that they have found it way they have found the inherent bias that exists, and the inherent prejudice against them. It has deeply affected them in terms of the science they wish to do and their access to opportunities in the field of academia. Next comes recognition between mentor and student. Of course, uh, a mentor, the power dynamics between mentors and students, of course, plays a very important role. For example, we'll come across certain examples in which a mentor has, mentors have because of their power and because of their position, the way they have made it difficult for their students or their mentees, and how that has affected the field also, and so on. So, yeah, all of these ethical issues continue to plague us in different ways, and these are true not just for neuroscience, and these are actually ethical issues that span across the different fields of science, and we need to be aware of these issues and ensure that we are not we are meeting the required ethical standards in our interactions and in our approach to science. This we have discussed that the 1950s are regarded as the golden is regarded as the golden decade for neuroscience because of the sheer number of discoveries that happened. There was a dramatic increase in the number of extremely important discoveries that happened during this decade, and it opened up our understanding and opened up the field of neuroscience for uh, several, like as a it made neuroscience as a field emerge much more strongly because of all of these extremely important discoveries, many of which we will be discussing in the upcoming video lectures. So we have already discussed regarding the double helix, the structure of DNA in great detail while we were discussing the assignment questions. In case you have any specific doubts, I can address those in the discussion forum. We have discussed about the fact that William Bateson was the one who introduced the term genetics in 1906. Thomas Hunt Morgan was the one who gave the gave this first strong evidence, experimental evidence for the chromosomal theory of inheritance, and mutations were mutations can be induced by X-rays was proposed by Hermann Muller in 1827, and genetic recombination was shown by Tatum and Lederberg in 1946, and the structure of DNA was, the idea that DNA is the genetic material came from these important experiments. So, first was the experiment by Frederick Griffith, in which he showed that DNA is the he showed the genetic transformation in Streptococcus pneumoniae. Then came the experiments by Avery, MacLeod, and McCarthy, in which they showed that DNA is the transforming principle, and they gave evidence that DNA might be the genetic material. But many people were not convinced, and that's when the Hershey and Chase experiment came in, the one with bacteriophages, in which they showed that DNA is indeed the genetic material. Then we have discussed the race to DNA, and how the institutional support played a very important role in the research environment of the 1950s, because the funding was given directly to the PI. It was not given to the head of the department, and there was therefore more opportunity for younger scientists to compete with each other, and the funds were given with few strings attached. They were given more freedom to pursue projects, and there was also a relative increase in funding as compared to previously. And so here we see the model that was constructed by Watson and Crick. So this is James Watson, and this is Francis Crick, who's pointing to the double helix structure that they have made. And like I explained, they did not themselves perform any experiments. They collected the information that was from the experiments that were being performed by other scientists. So they collected the early crystallographic data on the DNA structure that was carried out by Rosalind Franklin, which Morris Wilkins showed to them. And they came up with this model of two complementary intertwining helices of matching base pairs. So there's going to be the purines and the pyrimidines, basically the nucleotide bases, which are going to project from the sugar phosphate backbone. And this they published in a short article in Nature in 1953, titled Molecular Structure of Nucleic Acids, a Structure for Deoxyribonucleic Acid. And this paper was extremely famous for its last sentence, in which they suggested that because of this specific pairing for DNA, it suggests that there is a possible copying mechanism for the genetic material. So this is some information regarding the Watson and Crick model for the structure of DNA, regarding the size of the, the step size of the residues, and the angle of the residues, and so on. So so it slowly came to our understanding that DNA might be of different types depending. So there is A DNA, there is B DNA, there is Z DNA. So all of that information came about slowly. We have also discussed about DNA replication, which is basically the copying of the DNA, or the formation of new identical daughter DNA strands from an original parental DNA molecule, which is basically how the DNA undergoes replication and acts as the basis of inheritance. Then we discussed about how RNA was discovered in the glandular cells by Garnier in France in 1900, and it was found in the microsome fraction when he discovered this. And later it was understood that this RNA is required for the synthesis of proteins. So the process of synthesis of mRNA from DNA is transcription. The process of synthesis of proteins from mRNA is using mRNA as a template is translation. Transcription happens in the nucleus in eukaryotes, and translation happens in the cytosol. And the central dogma hypothesis says that the information passes from the DNA to the RNA to the protein, and once the information is passed to the protein, it cannot get out again. Basically, it cannot be reversed. Then we discussed about the discovery of polynucleotide phosphorylase by Oko and his colleagues, for which he got the Nobel Prize. So we know that the replication of DNA happens during mitosis, but in the adult brain cells, the neuronal cells are considered to be post-mitotic or terminally differentiated, which means that they do not undergo mitosis after they have become uh, mature cells. So, in those cells, there is no DNA replication which is happening. We still do not know the reason for that. Many different hypotheses do exist, and people are still trying to understand whether there might be a population of neuronal cells which present in the brain, which whether they are capable of uh, replication, or whether they can be made to be capable of replication in certain contexts, for example in case of injury, and maybe help in regeneration and so on. These are some ideas that are still they still continue to be explored. Then, regarding Barbara McClintock, who discovered the presence of mobile genetic elements, or famously called jumping genes, and whose work was not immediately accepted. She used the Indian corn or maize to do her experiments, and initially her work was not easily accepted. It took several years for her work to be uh, accepted by other scientists. Then we discussed about chromosomal abnormalities such as sickle cell anemia and several other genetic defects which might exist, such as Down syndrome, which is associated with trisomy of chromosome number 21. So trisomy means there are three basically, and there's an extra chromosome which results in the presence of results in this particular chromosomal abnormality known as Down syndrome. So this is the sequence of events that took place in 1953. The structure of DNA was first a model of the structure of DNA was discovered in 1953 by Watson and Crick. Then by 1963 the genetic code was established. Then in 1970 the recombinant DNA technology and the beginning of DNA sequencing started. In 1980 the polymerase chain reaction was invented. Then in 2000 the first the sequencing of the full human genome was finished. So this is the famous article by Watson and Crick, in which they have in which they have published their model for the double helical structure for DNA. I strongly encourage you to go and read this classic paper. So, with that, I come to the end of this session. Thank you very much for listening. If you have any questions, feel free to drop them in the discussion forum. I would be happy to answer you. Thank you very much.